Now, sometimes we need to make an identical copy of our DNA. Like for example, if you have a cut or a scrape, we need to make more skin cells. Well, to make a new skin cell, you need to make a copy of an existing skin cell. Okay, so the way you make an existing copy of DNA is we need to, first of all, split it in half. We have an enzyme called DNA helicase. Because it's a helix, ASC is an enzyme. All enzymes are proteins, and proteins speed up chemical reactions. So DNA helicase is going to unwind and unzip our DNA. Just like this. I'm going to spread them out a little bit farther apart, and you'll see why in a minute. Okay, but these strands all stay together. That sugar phosphate backbone remains unbroken. We're simply unwinding and unzipping. It's like having a hoodie and undoing the zipper from the top down. Okay, that's the job of DNA helicase, to unwind and unzip. Then what we're going to do is we're going to put this guy away for a little bit, and we're going to get another enzyme called DNA polymerase. It's an enzyme because it ends in ASE. It's also a protein because all enzymes are proteins. The job of DNA polymerase is to polymerize these monomers into a polymer. So you're going to make a brand new DNA molecule. And for this case, so it sticks out, we're going to use these shaded purple ones. So basically, the foods that we eat most of the foods that we eat are from living tissue, other than something like salt on your French fries. Salt, NaCl, does not come from cells. It's not living. But when you're eating an apple, when you eat lettuce, meat, that's coming from living tissue, which contains cells, and all cells contain nucleus. Well, most of the cells that we eat contain a nucleus, and inside a nucleus is DNA. Your body breaks down those nucleic acids, that DNA, into nucleotides. So you basically have a whole pile of spare parts in all your cells. And it's a good thing because we're going to use these spare parts to polymerize our original strand and make two strands. Okay, so we're going to look at our spare parts here, and we are going to make a new strand. So matching up with C, we need to find a G. There's a G right there. So DNA polymerase is going to stick a G on there. It's going to stick an A over here to match up with that T. It's going to stick a T over here to match up with that A. It's going to stick a G over here. And it's going to put a C over here. And it's going to put a C over here. And an A over here. And an A over here. It's going to do the same thing down this strand. So we're going to plop a C right there. We're going to plop a T right there. We're going to plop an A right there, because that's complementary. You're putting the complementary base and you're binding them up. There's actually another enzyme called DNA ligase. I don't have it here, but what ligase does, ligase actually sticks these guys on together, glues them together. Let's see, we need a G right there, and we need a G right there, and we need a T right there, and we need a T right there. So now the job of DNA polymerase is done. DNA polymerase is done. DNA ligase comes in, glues these guys together, and now we've got two identical strands. If you look at them, this strand on the left is identical to this strand on the right. The only difference is that the original strand got split in half permanently, and that was the white strand here. The strand built out of spare parts is here, but literally, functionally, these are identical, okay? We call this semi-conservative. The reason we call it semi-conservative is when you conserve something, you save it. So we saved half the original strand. So now you've got 2 identical strands of DNA. We do this again, you end up with 4, again, 8, again, 16, again, 32, 64, 128, 256, 512, 1024, 2048, on and on and on. That way we can double our DNA in no time flat, okay? And then we take DNA helicase and we rewind and re-zip, hydrogen bonding here, and that's it. We call this DNA replication.